The secretory cells present in salivary glands are of two types, the serous and the mucous cells. These secretory cells, also called acinar cells, are housed in secretory end pieces. The secretory end pieces are continuous with the main excretory duct of the salivary gland through intermediary ducts. The lumen of the end pieces open into the intercalated duct, which is continuous with the striated duct, which in turn opens into the excretory duct of the salivary gland. Also note that the ducts are lined by ductal cells. Now the secretory end pieces and the intercalated ducts are associated with stellate-shaped contractile cells called myoepithelial cells. They are located between the basal lamina and the secretory or the ductal epithelial cells. They are also called as basket cells because they look like a basket holding a secretory unit. The myoepithelial cells have a central body with cytoplasmic extensions embracing the acinar cells. These cells, however, are more fusiform shaped and have lesser cytoplasmic extensions when associated with the ductal cells. The cells are called myoepithelial due to the fact that they are primarily derived from epithelial cells but exhibit certain characteristics of smooth muscle cells. Myoepithelial cells have a central body with 4 to 8 cytoplasmic processes. These processes may further branch out to secondary and tertiary processes, thus securely hugging the acinar cells. They have a flattened nucleus in their cell body with cytoplasmic organelles around it. The plasma membrane of the cell appears to run parallel to the membrane of the adjacent acinar or the ductal cell. The plasma membrane of the myoepithelial cell has small invaginations called caveolae. The myoepithelial cell is attached to adjacent acinar or ductal cells through desmosomal junctions. Now, it has to be remembered that myoepithelial cells have smooth muscle-like properties but are primarily epithelial cells having an ectodermal origin. However, they also harbor cytokeratin filaments and desmosomal junctions, both characteristic of epithelial cells. The main function of the myoepithelial cell is to contract and help squeeze out secretory products from the secretory end pieces to the ducts and finally to the oral cavity. So these repeated contractions of myoepithelial cells help in maintaining patency of the ducts. Myoepithelial cells have also been postulated to maintain the structural integrity of secretory end pieces and the cell polarity of secretory acinar cells. Interestingly, studies report that myoepithelial cells have a protective function in that they may secrete certain proteins that may have anti-cancer effects, like a tumor suppressor activity and an anti-angiogenic activity. The tumor suppressor activity refers to inhibition of growth of cancerous cells, while the anti-angiogenic activity refers to the inhibition of proliferation of blood vessels, which may be a source of nutrition for cancer cells. Another controversial and speculated function of myoepithelial cell is the transport of metabolites through the multiple membrane enfoldings called the caveolae. Myoepithelial cells can be identified through numerous stains and methods like hematoxylin and eosin stains, fluorescent stains, special stains like silver staining, and immunohistochemical markers like alpha SMA, cytokeratin 14, cytokeratin 17, calponin, integrin, and P63.